All right, so it is 2 o'clock and we will get started here, and I will put up the PowerPoint. All right, so for biochemistry, what we're going to do for the first couple sessions is just a massive review of fundamental organic chemistry and biology. So hopefully all this won't be new to you, but we're going to go probably way faster. Well, definitely way faster than you learned it in your other classes, just because this is review. So we're first starting with the different functional groups and linkages that are commonly used in biochemistry. Now, unlike organic chemistry, where you can have a lot of different molecules, biochemistry, we mainly focus just on a few types of elements: carbon, nitrogen, oxygen, sulfur, phosphorus, because those elements are found in living organisms. While in organic chemistry, you could use, just about any molecule on the periodic table that you want. So for these tables of functional groups and linkages on this page and the next. So this is what I'm going to have you do before test 1. And this is one of our first big rote memorization things. There's no way around it. We have to know these structures. So what I want you to do from this table is I want you to know the information in this table. That is, when test 1 comes, what I'm going to do to you is I'm going to give you one of these structures, right? And then you'll have a multiple choice question where it says, what's the name of this structure? You're going to have a couple questions like that. So I want you to be able to know the names of the functional groups and linkages in these two tables, this table and the previous table. The reason for that is it just makes life a lot easier once we start to look at enzymes and more complicated systems. If I can say, like, look at this alcohol group, or it's going to react with the aldehyde, or it's going to form an ester during this mechanism, that's going to make life a lot easier. So to be able to actually talk about biochemistry at a higher level, we need to have the correct vocab. And one of those vocab things start with these functional groups. So start working on those. I would suggest, like, flashcards, work a little bit at a time, 5-10 minutes a day with those flashcards. And you should have those memorized, no problem, by test 1. But just to let you know, 100%, I'm going to ask you a couple questions where I give you the structure and you have to tell me the name. All right. So from the videos I have, and this will be a theme throughout our whole course for these in-class sessions, I'm going to be asking you questions, right? And these questions are a way to test your understanding and for me to measure the understanding of the class during these live sessions. Now, these questions aren't graded in any way. They're mainly for your benefit and also for my benefit to know where we're at, to see, how you're progressing and how you understand the material. And they're also a great way to review when you're looking back at your notes, studying for the test. You will know how well you're going to do on the test basically by how well you can answer these questions. Can you answer all the questions? If yes, you should have no problem on the test. Are there questions you don't know? Well, that might show up on the test. And if you don't know it in the practice, you're not going to know it for test time. So here's some rather simple questions. So can anyone tell me, if we're talking about dry weight, what is the most abundant element in humans? And for B, what is the most abundant molecule in our body? Carbon. That is correct. So if you take all the water from our body, we're mostly carbon. We are carbon organisms. Every single molecule we look at this semester is going to have a carbon backbone. So yeah, so that's carbon. Now, what molecule is the most abundant in our body then? You want to have any ideas about that? Water. Correct. We are more or less big old water balloons. Roughly 70% of your cells are made out of water. So yeah, we are water carbon organisms. So when we talk about the chemistry portion of this class, one of the first things we're going to talk about is how water has an influence on biochemistry. Basically, without water, there is no biochemistry. And outside of water, the other main thing we're going to talk about is carbon chemistry, which is why organic chemistry is a prereq for this class. In organic chemistry, that is carbon chemistry. And we're going to use a lot of the ideas from organic chemistry to understand how biochemistry works. So just to get an understanding of our functional groups, I have questions like these. Again, these are to test your understanding. And I do have polls. Since this is an online class, I want to try and get user participation. And so I have these Zoom polls. Now, just to let you know, like I said, these Zoom polls aren't graded in any way. And in fact, I can't even see how you answer individually. These are completely anonymous. All I can see is percentages. And also for these polls, if they get in the way of you answering, what you can do is minimize the poll, then go back to the top of the page and click on the polls again, and it should pop back up. But what I want you to do is see how well is your knowledge from organic chemistry, and tell me, from this simple molecule, which atom is showing us a carboxyl group? So take a minute and see if you can answer that. All right. Looks like we have most of the people in, and most of you got it correct. So the carboxyl group is 5, right? So C double bond O, OH, that would be our carboxyl group. If I asked you for just a carbonyl group, that would be 1. So this is a carbonyl. When you add the OH, that becomes a carboxyl. So again, this is another good example of how I would ask you on the test for that table of functional groups. I could ask you a question like this, where I say, here's a molecule, I've numbered the carbons, which one is this functional group? So just another way I will probably test you on test 1. All right, so in biochemistry, we're going to talk a lot about polymers, because that's basically what biochemistry is, a lot of it at least. It's the chemistry of biopolymers. And we're going to look at 4 different polymers this semester. We're going to look at proteins, nucleic acids, polysaccharides, and lipids. And to really understand the polymer, we need to understand the basic unit, which is a monomer. So for proteins, that's amino acids. For nucleic acids, that's the nucleotide. For polysaccharides, that's a monosaccharide. And for lipids, that's a fatty acid. And this picture on the right is just kind of showing an idea of what we're going to look at, how we have a polymer, and from that polymer, we can get more complex structures. But yeah, this semester, we're going to look at all 4 of those polymers in depth as one of our goals. Now, just to continue, oh yeah, question here. Fun, I think so. So hopefully you've seen these polymers at some point in your educational career. So again, just as a refresher, just to test yourself, I want you to tell me this picture that we're looking at, what class of molecule do you think we're looking at? Is this a lipid, a carbohydrate, amino acid, or is this a nucleotide? So we'll go about like 10 more, 15 more seconds. And if you're not sure on these, always feel free to give a guess. Like I said, I don't know how individuals answer, and you're not getting credit taken away from you. All right, so let's take a look at this. And most of us got this right again. This is an amino acid. So how can we tell if what we're looking at is an amino acid? And we will talk about this when we get to the amino acid section. But for amino acids, you are always looking for this part of the molecule. This is called the backbone of the amino acid, and it never changes. Amino acids always go NH3, then what's called the carbon alpha, and COO minus. The rest of the molecule, this is called the side chain. And it varies. For organisms here on Earth, there are 20 common amino acids, or if you talk to some biologists, they'll say 21, but I say 20. And we're going to learn throughout this semester the structure of all 20 amino acids, and we're also going to look at the function. But to determine if something's an amino acid, just look for that backbone structure. How about this molecule? What do you think we're looking at here? Let me get that pulled off. This big old long molecule, what possibly could this be? Is this an amino acid, a lipid, a carbohydrate, or a nucleotide? All right, seems like we have just about everybody in. All right, so here we got our first discrepancy. 50% said a lipid, 50% said a carbohydrate. So the correct answer here is actually a lipid. Okay, how do we determine if something's a lipid or something is a carbohydrate? The main thing is lipids have very long carbon chains. And these carbons are without hydroxyl groups attached to them. generally lipids only have like one hydroxyl group or one polar group at the head. The rest is just carbon. Carbohydrates, on the other hand, for the most part, almost every carbon, so almost, will have an OH attached to it. And that's actually in the word, right? Hydrate, OH, right? Water, hydrate. Carbo, carbon. Carbohydrate means carbon water, right? So that's how we can tell if we're looking at a lipid or if we're looking at a carbohydrate. So any questions so far on anything I've gone over up to this point? All right, if not, we'll move on. So continuing our tour of what we should have learned in biology, hopefully. What is this? What is this lovely molecule? This is a carbohydrate, a lipid, amino acid, or nucleotide. All right. Looks like we crushed that one as a class, so good job, everybody. This is a nucleotide. So what to look for in a nucleotide? A nucleotide always has a sugar. Then it has some nitrogen base. And it'll have phosphates connected to it. Unless it's in a chain, then it'll only have one phosphate. It can have like 3 phosphates. By the way, testing your biology. Am I looking, or are we looking at RNA, or are we looking at DNA right here? Extra bonus question. RNA, that is correct. We'll get into this in the nucleotide chapter, but this carbon right here will tell you if it's RNA or DNA. RNA has an OH. DNA has a hydrogen because D is deoxy, right? So that means it's lost a hydrogen. Yeah, very good. That's what a typical nucleotide looks like. How about this? We're looking at a different representation here. So this one's a little more tricky than the rest. But what class of molecule are we looking at now? Just to give you some representation, red is oxygen, blue is nitrogen, green is carbon, white is hydrogen. So see if you can use this, what's called a ball and stick representation, to determine what type of molecule this is. And as always, when doing these, feel free to ask me questions if you'd like. But we'll go like another 20-ish seconds on this. All right. So what we're looking at here is an amino acid. Looks like the majority of us got that as well. So remember what I told you for amino acids. Always look for a nitrogen connected to a carbon connected to a carboxylic acid. And that's what's being shown right here. During this course, we will be looking at molecules with different representation like this because this has very useful information. What this is showing us is the charge density of this molecule, right? So red would be negative charge density. Blue would be positive, and this green or white, that's neutral, right? And this is, having this type of representation is very important when it comes to molecule-molecule interaction. In biochemistry, you usually don't interact via covalent bonds. It's all non-covalent interactions. So hydrogen bonds, ionic bonds, hydrophobic interactions. And so to really understand how molecules interact, we have to have a good idea of what kind of charge surrounds the molecules, right? This is very important in drug design as well. So let's say you have a target protein and you want to inhibit that protein or enzyme. Well, you usually want to inhibit a specific part of that enzyme. And so to make a drug that will interact with that enzyme, you need to know the charge character. So for example, if the part of the molecule you want to interact has like a positive, and a negative charge next to each other, your drug of interest should have a positive, negative charge spaced out for that interaction, and then it'll fit. But that's the basics of drug design in 2 seconds right there. But yeah, this is an amino acid. Any questions about this representation or anything at all up to this point? All right. So not too many more of these. I think this might be my last one. What is this? What are we looking at here? What is this molecule? I already kind of told you what it was earlier as well. Do about 10 or so more seconds. All right, so this is a carbohydrate. Remember, when we're looking at carbohydrates, it means carbon hydrated. So carbon hydrate. Also, remember from your organic chemistry, when we're looking at straight lines like this, that represents carbon because chemists are too lazy to draw like C, 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 when you can just go da, 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 da. So whenever we see solid lines, the little bumps in the lines are carbon. So don't forget that from your organic chemistry. All right, so that was your, I don't know, 10-minute crash course on, remember this chemistry from previous classes? Now we're going to do the 10-minute crash course of, do you remember biology at all, right? Because this is biochemistry. We are marrying the fields of biology plus chemistry. We're looking at the chemistry of life. And the main types of systems we're going to look at is prokaryotic and eukaryotic. We're going to bounce around between the two. Prokaryotes are easier to study, but eukaryotes are more relevant for us because we're eukaryotic. So just to make sure we remember, here's another question that we should have learned in first year biology, hopefully. So good way to refresh yourself. During the evolution of eukaryotic cells from prokaryotic cells, what developed? What was created when eukaryotic cells evolved from prokaryotic cells? All right, seems we have most people in. So let's take a look at this. DNA is the instruction guide for a cell. So we've always had that. So that's not it. The cell membrane surrounds the cell. So if you don't have a cell membrane, you don't have a cell. That's not it. Ribosomes. Ribosomes make proteins. You can't have a cell without proteins. That can't be it. Therefore, E can't be it. So the correct answer is the nuclear membrane. So in a prokaryote, if you remember, the prokaryote has its DNA all diffused throughout the cell, while a eukaryote has a nice little nucleus that our DNA is all tucked into. Haven't taken biology since high school? Well, we don't go that much into biology, but it's ever present in that everything we look at is inside of a cell. So I would say to just remind yourself of biology, what we need to know is the major differences. Whoops, come on, virus. Not right now, please. Sorry, my antivirus just popped up. What we need to know is like, what's the difference between a prokaryote and a eukaryote? And some of the differences are one, nuclear membranes, organelles, right? Eukaryotes have organelles, prokaryotes don't. Size. Generally, a eukaryotic cell is much bigger than a prokaryotic cell. But when you're talking on a biochemistry level and looking at individual proteins, a lot is the same between prokaryotes and eukaryotes. And we're going to talk about that, how information we learn from prokaryotic cells, it's roughly applicable to your eukaryotic cells. Proteins function the same way in both organisms. Okay, a couple more questions to refresh your memory on biology. I know this feels like giant quiz time, but this is a good way to, when you go back to study for test one, can we answer these? So for eukaryotic cells, where do we get energy from? And when we talk about eukaryotic cells, which of these statements is not true? right? Seems like we're mostly in. And it seems like we do have a pretty good knowledge of our biology. Very good. So just to go through the different types of organelles or locations in a eukaryotic cell. ER, endoplasmic reticulum. This is where protein processing can happen. That's where it happens somewhat. Also on the ER, if it's rough ER, that has ribosomes and it makes protein. So the rough ER is the site of protein production. As a reminder, there are free ribosomes as well. So not all ribosomes are on the rough ER. There are ribosomes just floating around in the cell. That's what's shown here in this picture too. You have these little black circles. Those are supposed to be free ribosomes. Nucleus, DNA. That's the instruction manual for the cell. That's the brain of the cell. All of our DNA is going to be contained in our nucleus. Mitochondria, powerhouse. This is where ATP synthesis happens. I'm sure you heard this in other classes, but the current theory is that mitochondria used to be a separate cell, and then early eukaryotes just kind of like ate it, but didn't destroy it, and used it to produce energy for it. Some evidence to this is that the mitochondria inside your cells, they divide independently of our cells. So they'll divide on their own. Their DNA is a circle. Prokaryotic DNA is a circle. So it has the same DNA structure as a prokaryote, while eukaryotes are not circles. So just some evidence in there that mitochondria used to be prokaryotes. Peroxisomes, they break down things. So peroxisomes can break down fatty acids, and they can also break down some proteins. But mostly peroxisomes are used for fatty acids. Golgi apparatus, that is protein processing / shipping, right? Any protein that needs to go to the cell membrane, they have to go through the Golgi apparatus first. And we'll go into depth on that whole mechanism, how we go from ribosome to Golgi to cell membrane. So B, which of the following statements is true or is not true? Well, organelles, yes, that's what makes a eukaryote. We have systems inside of our cell that are their own little like factories. And they're separated by a membrane. Mitochondria, peroxisome are all types of organelles. Usually 100 to or 10 to 100 microns in diameter. So what's a micron? Better known as a micrometer, or 1 micron is 1 times 10 to the minus 6 meters. And that's true. When we talk about biochemistry, we're talking in very, very small sizes, right? Because we're going to be talking about proteins, DNA, sugars. They all fit inside of a cell. So the unit of distance is way tinier than a micron. Usually when we talk about biochemistry, the units that are normally used are the nanometer, which is 1 times 10 to the minus 9 meter, or a special unit, the angstrom. An angstrom is 1 times 10 to the minus 10 meter, or the length of a carbon-hydrogen bond is 1 angstrom. So that's true. Either unicellular or multicellular. So unicellular could be yeast or amoebas. Multicellular are us. So that's true. Archaea, that's the false one. So there are 3 kingdoms, or yeah, I think I have my organization right. 3 kingdoms. You have your prokaryota, your eukaryota, and your archaea. So archaea are a completely different system of life than the eukaryota. We will hardly ever talk about archaea in this class. Archaea biochemistry is really different when it comes to eukaryotic. There is some overlap, like I said, for proteins, but there's also a lot of differences. And so to keep it simple, we're mainly going to focus on the eukaryota and the prokaryota. What's the answer to the first question? That would be the mitochondria. That is where we do aerobic metabolism. That's where we make our ATP. All right. And here's what I was talking about. So we have our bacteria, our archaea, and our eukaryota. This is the evolutionary tree of life. I will be talking about evolution a lot in this class. So just be aware of that fact. What I'm looking for you is for you to understand evolution. Do you know the scientific basis of evolution? Do you know the logic behind evolution? That's what I'm looking for. And so tree of life, basically branches closer to each other have a more common ancestor. So when branches meet, that node is a common ancestor. The further you are away from a specific branch, the further you are in evolution terms. So animals up here, we're actually slightly closer to fungi than we are to plants when you look at evolutionary scales. And eukaryota itself are closer to the weird archaea than we are to the bacteria. And when we get into evolution, it'll mostly be protein evolution. We're going to see how proteins have changed over time and what tools we can use to monitor that change. So from what you've learned of evolution, what best describes evolution? So see if you can figure out which one best describes our theory of evolution. So which one best describes evolution? All right, seems like most of us have answered. And the correct answer is actually, let me get my pen out, is E. It's all three of these. So evolution, there's no end game for evolution, right? Like humans aren't the pinnacle of evolution. Evolution just happens. There's no goalposts. It's adapting to changes. It's fitness of populations, right? So when we're talking about evolution on a big scale, we're looking at populations. So they're not shifting towards like, I'm going to get this goal. It's just what challenges are present in my environment, which organisms can overcome those challenges and propagate their genome. And over time, the population changes. And evolution is ongoing. Evolution's like never stopping, but it's constrained by its past. What we mean by that is that for evolution to happen, you have to build on something. So when we talk about evolutions of proteins, right, we need proteins to be able to build on. So it's very hard to come up with like a brand new protein. What happens is that you have an existing protein and it gets changed somehow. And for that change to happen, you need sloppiness. By sloppiness, we mean errors. If our DNA replication was 100% perfect, we never made any changes to our DNA during replication, we would never evolve. For evolution to happen, we need to make mistakes. Errors need to happen. For us, errors happen very small detail. It doesn't happen often because if mistakes happen too much, we would basically die. We would have too many errors. We can't make the proteins we need. But we do need some sloppiness for this to happen. One good example of this that I just thought off the top of my head here are cancer cells. The way cancer cells propagate is through sloppiness. When a cancer cell really starts to grow, that's because they turn off a lot of the safeguards that prevents them from growing inside your body. A lot of the checks and balances we have for our own DNA replication, they just say that takes too much energy, that takes too much time. Just keep replicating DNA because I need to multiply very fast here. And when they do that, they get errors in their proteins. Some cancer cells will die while others will continue to propagate. Then when we start to treat the cancer with different like drugs, some of those cancer cells will be susceptible to those drugs and die off. However, because cancer cells are so sloppy in making and replicating their own DNA, some will become immune to these drugs because the proteins that the drugs are targeting can no longer be targeted due to a mutation. So these cancer cells have evolved and now they are immune to these drugs and they can propagate. Then we have to find a new type of drug. Another great example of this are bacteria. We have bacteria that are more or less immune to every type of antibiotic we have because they have evolved over time due to genetic changes that were completely due to random chance. They have become immune to these antibacterials, and it's a constant struggle in hospitals to deal with these superbugs. But any questions about just the very basic idea of the theory of evolution? All right. All right. So in the previous 40 minutes, we just went over everything you should have learned in organic chemistry and everything you should have learned in general biology. Now we're going to learn about, do you remember this from general chemistry? In this class, we're going to talk about thermodynamics of systems quite a bit. That was actually my specialty in grad school. I'm technically a biophysicist. So that's a biochemist who specialize in the physics of biological systems. And one thing of physics is thermodynamics. So we have to remember some ideas from thermodynamics. And the big equation that hopefully you remember from general chemistry is the Gibbs free energy equation. Delta G equals delta H minus T delta S. All right, so let's break these down. Delta G is energy, or the ability to do work. H is enthalpy, or heat. It's kind of the same thing. It's not a one-to-one comparison, but it's close enough. If enthalpy is negative, then it's exothermic. You can think of this as heat exiting. While if enthalpy is positive, it's endothermic. This is heat entering. So in exothermic reactions, heat leaves the system. In endothermic reactions, heat enters the system. Our other term here is entropy, that's delta S, or chaos. They Again, they basically mean the same thing. It's not a one-to-one comparison, but it's close enough to get the idea across. And the universe always goes towards higher entropy. So if entropy increases, chaos increases. It's more disordered. If entropy is negative, chaos decreases. It's become more ordered. And the universe is in a constant state of becoming more and more disordered. Every second that passes by, things become more disordered naturally. And life, us, we fight against that. So we try to make things ordered. So in our body, the majority of the energy, or just about all of the energy we use in our bodies, is to bring order to disorder. So delta G, if it's positive, it's endergonic, non-spontaneous. If it's negative, it's exergonic, it's spontaneous, and 0 is equilibrium. So let's go over these terms, spontaneous and non-spontaneous. Spontaneous means it will happen. So my pen, right? So I have my pen hovering above my desk, and I let my pen go, it hits the table. That is a spontaneous reaction. Delta G is negative for that. Now, right now, I have my hand over my pen, and I am trying to force with my mind my pen to go back into my hand. That is non-spontaneous. That will never happen. I could sit here focusing on my pen for the next 10,000 years, and that pen will never jump into my hand. So that's a non-spontaneous reaction. What an equilibrium means, an equilibrium, I can explain that best by having a reaction. Let's say A plus B goes to C. So a very simple reaction. A and B combine to make C, and C can break down to make A and B. Equilibrium means the forward and reverse reaction rates are equal. The speed of the forward and the reverse reaction rate are equal at equilibrium. Equilibrium does not mean products and reactant concentrations are the same. Equilibrium is not that. Equilibrium is talking about speed of reaction. The forward and reverse reaction rate is the same. When we talk about thermodynamics, which is the study of energy, that is separate from the field of kinetics. So kinetics is the study of reaction rates. And thermodynamics is the study of reaction energies. And these are independent of each other. What I mean by that is, let's say I have a iron nail, right? And I leave that iron nail at a beach. Eventually, that nail will rust. The delta G for this reaction is negative. But the rusting of that nail might take a few years. So it will happen. It's spontaneous, but it'll take a long time. Here's another, well, let's take the example I just did. Pen over desk. I let go of the pen, pen drops and hits the desk. This delta G is also negative. This happens in a matter of seconds. So although both reactions are negative, the time is vastly different. So you can never look at a free energy and tell me how fast it will go. That's impossible. You can tell me whether it will eventually happen or not, and that's spontaneous or non-spontaneous. And I think for right now, we'll leave it right here. Is there any questions about anything I went over today in our madcap recap? session of everything you've hopefully learned in previous classes. Right, if there are, well, looks like we have one here. Nope, okay. Well, if you do have questions over the weekend, please shoot me an email. Remember, I will now put up a homework on Wiley Plus. This homework will be due in a week. So it'll be due at 2 p.m. on the 29th. If you have any questions about that homework, please let me know. Anything else, please let me know. Not a question, but do we have a group chat or a group me for everyone? Blackboard, there's a message board, and I think someone on there set up like a group chat. I don't have one set up myself, but there is, I think somebody did try and organize that. If you want to ask general questions, that's not an email. You can also use the message board and I can see those. Do you have to download any software to take the exam in the future? You might have to, you might have to download an add-on for your web browser called ProctorU. I'm like 90% sure I'm going to use that for my biochem exams. But all that is an add-on, which means you need a computer and you need a webcam. Webcams you can get from the library, computers you can get from the library as well. But that should be the only software, and I'll have more instructions on that when we get closer to test time. Is there a link for that? There is, I don't have it on the top of my head. If you want to Google ProctorU, like student setup or student test, what that will do is it will walk you through the steps. And like I said, like a week or two from test time, when I'm 100% committed to ProctorU, I will have all that up, but I still want to leave my options open in case to look for other type of testing software. All right. So that's all I have for you for today. So make sure you do your homework. Make sure you complete the reading guides for Monday, and I will see everybody hopefully on Monday. Have a good weekend, everybody.